This is part 2 of our discovery of chapter 9 in our Human Anatomy and Physiology text on muscle anatomy and physiology. In part 1, we homed in on the microscopic anatomy of a muscle cell, and now we can take a look at some more dynamic things happening in the cell, namely the presence of an electric charge on the membrane. Neurons and muscle cells have a resting membrane potential, it's called. We'll also talk about excitation of a muscle cell, and that involves changing that electrical charge on the membrane momentarily, and then that will introduce some changes inside the cell called excitation-contraction coupling. And indeed, the ultimate thing that we want to see happening is contraction of the cell. Cross-bridge cycling is a mechanical series of biochemical events and converted to mechanical events that will cause the cell to contract. All right. Let's check it out. Resting membrane potential. So these cells have an electric charge on the membrane. And how does that come about? Well, there's two things that sort of contribute to the presence of that imbalance in charge. Negative on the inside, positive on the outside of the membrane. Firstly, the sodium-potassium pumps found in the membrane of all your cells are constantly pumping sodium ions out of the cells and potassium ions into the cells. And that, in conjunction with the kidneys, which regulate the concentration of those ions in the extracellular fluid, produces concentration gradients. So think of it. If potassium ions are being pumped into the cell all the time, they accumulate in there. There's a high potassium concentration inside the cell, and the kidneys keep a very low potassium concentration outside. The pumps are continually pumping sodium out. And so all your cells have a low sodium content in their cytoplasm, and the kidneys maintain a high sodium and chloride concentration outside the cells. That's the predominant solute in the fluids in your body, extracellular fluids in your body. So we have these big concentration gradients or differences. So that's a force that can drive diffusion, a potential to drive diffusion. The second thing we need to note is that at rest, the membrane has what are called leak channels. The membrane is slightly more leaky, where can I go, for potassium than sodium. That just means that potassium ions can trickle across the membrane at a slow rate. The sodium pumps keep the gradients constant, but some potassium ions can trickle out of the cell, and as they do so, they leave behind a net balance of negatively charged substances or chemicals. Let me show you a little cartoon drawing I made that maybe will help illustrate what I'm talking about. Think of a cell that we're going to start it up like starting up your lawnmower. It's just a cell with nothing happening in it yet. It's just a cell membrane full of positively and negatively charged chemicals, ions, proteins that have negative, a lot of proteins in the cytoplasm have a net negative charge. Their side chains sticking out of the backbone of the protein have negative charges. So we got all these positive and negative charges, and we essentially have equal numbers, let's say. We have a balance. Then we'll start up a sodium-potassium pump in the membrane. And as the sodium-potassium pump operates, we're going to deplete the cytoplasm of sodium, but have lots on the outside. And I use the size of the symbols to illustrate concentration. So a big Na here represents a high sodium concentration. A little Na means a low sodium concentration. And also the pump is pumping potassiums into the cell, and they accumulate. So we have a big K representing a high potassium concentration inside and a low concentration outside. So those are driving forces that can move those ions through a process called diffusion if there's a way for them to cross the membrane. Remember, these ions can't cross the phospholipid bilayer. They can only cross if there's a protein present that can open up a way for them. Well, there are such proteins in the membrane at rest. This is still a resting cell. The sodium pumps are working, creating those gradients, and the membrane lets potassiums trickle out. Some sodiums can trickle in occasionally, but it's about 100 times easier for potassiums to leak out than for sodiums to leak in. So we started out with a nice even balance of plus and minus charges. Now we're letting some positive charges escape, and the result is we're going to have a relative excess of negative charges inside the membrane. That is the resting membrane potential or charge. It's like a little battery. There's negative charges inside and a net excess of positive charges on the outside of the membrane. If we measure the actual value of that charge, how much charge is there across the membrane? Charge is measured, or charge difference is measured in volts, and it's minus 70 millivolts, meaning minus charge inside. That's a significant charge separation or difference across the membrane. That's what we mean by a resting membrane potential. The word permeability means the ease with which something may cross the membrane. In other words, if a membrane is permeable to potassium, that means it can cross the membrane. It's a matter of degrees. At rest, the permeability for potassium is low, but it's still 100 times higher than sodium. We're going to encounter a situation in just a little bit in which the permeability changes dramatically and becomes high for sodium and high for potassium. But at the moment, at rest, these permeabilities are low, but more so for potassium. All right, that's how we get a resting membrane potential. So let's say we have this idea that we want to move our skeleton. We want to contract a muscle to do so. So we send a signal, an impulse it's called, down a spinal cord motor neuron. A motor neuron is just a neuron carrying information out from the spinal cord to the muscle. That's how we get the attention of the muscle. Send an impulse down that motor neuron, it's called. And the motor neuron, that impulse is called an action potential. What it is it's a momentary change from the negative resting membrane potential to a positive interior of the cell and then back. And what happens is that momentary change in the polarity of the potential in the membrane sweeps down the membrane like a wave. In this case, it's sweeping down the axon of the motor neuron, and eventually it's going to trigger such an event in the muscle cell. So that action potential will essentially jump from the motor neuron over to the muscle cell and induce an action potential there through the action of a neurotransmitter. So let's take a look. Here's a little cartoon illustrating the thought process of contracting a muscle. We want to contract a muscle to do some task. And so an impulse or an action potential will spread like a wave down this axon of a motor neuron. And you can see it has little branches and little contact patches with the muscle cells. These are all the muscle cells, and that motor neuron is going to activate the muscle cells so that they will contract. Those points where the motor neuron comes into the vicinity of the muscle cell is called the neuromuscular junction. Right? We have a neuron and a muscle cell and a junction. When the impulse or change in polarity of the membrane potential on the axon sweeps down to the terminal, this is called, there are membrane channels that are sensitive to that change in polarity. When the action potential arrives, calcium channels open in the axon terminal and calcium ions diffuse, they rush into the cell. I don't know if you remember, but we said in the last section that the cytoplasm of the cell, whether in this axon terminal or in the muscle cell, is vanishingly small in calcium ions. There's 1/10 millionth molar calcium in those cytoplasmic fluids, so in essence none. But there's 9 millimolar calcium in the extracellular fluid. That's a huge concentration difference. So calcium ions rush into the cell by diffusion, by virtue of their just random energy, and that rise in calcium concentration triggers exocytosis. And the little green balls and all these vesicles you see stored in the axon terminal is acetylcholine. Acetylcholine molecules diffuse that across this narrow space between the cells called the synaptic cleft and bind to their receptors on the muscle cell membrane. So the little green balls are binding, the acetylcholine is binding to its transmembrane protein receptor. Now it turns out that those receptors are also ion channels. They're switchable ion channels, just like we saw in the last section when we talked about ligand or chemically gated channels. These channels have been opened. They've been switched on, and now they can let ions pass through. If the acetylcholine was not present, how would they look? We'll see in a second. So here's a close-up look at one of these acetylcholine receptors. And it turns out that the acetylcholine receptor is a nonspecific channel that allows both sodium and potassium to cross the membrane. And we saw that there's a concentration gradient for both of those ions. And what's the net effect going to be? A lot more sodiums are going to rush into the cell than potassiums are going to leak out. Why is that? Well, it turns out that like charged species repel each other and opposite charges attract. And so if you imagine, we know that there's a high concentration of sodium outside the cell and a low concentration inside that already represents a driving force for sodium to diffuse in. But the positively charged sodium ions are also strongly attracted to the negatively charged interior of the cell. So two forces operating on those sodiums to drive them into the cell, and they rush in with alacrity. Potassium ions are more highly concentrated inside the cell, which represents a driving force for diffusion out, but yet the potassiums are positively charged, and they're attracted to those same negative interior charges, so it's more difficult for them to diffuse away, to escape from that attractive interior. So less potassiums go out, more sodiums go in. So we won't probably mention that again. We'll just remark on the fact that when we open up these channels, sodium ions rush into the cell. But you'll know the technical fact that some potassiums are going out as well. Okay, so what happens when we let a bunch of positively charged ions into an interior of a cell that was negatively charged? Now this area is not negatively charged anymore. There's not a net excess of negative charges. And we call that depolarization. Right? Polarized when there's a negative charge inside and a positive outside. Now we've added some positive charges and depolarized the membrane. A quick aside before we go on and see what's going to happen as a result of that depolarization. If we decide to stop contracting the muscle, in other words, we stop sending impulses down the motor neuron, stop exocytosing acetylcholine, the contraction will be ended immediately, because in the synaptic cleft resides an enzyme called acetylcholine esterase that very quickly destroys any acetylcholine that's present. So when we stop releasing acetylcholine from the axon terminal, it'll be quickly destroyed and everything will go quiet. We'll go back to the resting conditions. So this is just a little cartoon visualization of an acetylcholine molecule being broken down to an acetyl group and a choline molecule. Those no longer can bind to the receptor. And those can be taken up by the axon terminal and recycled, restored back to acetylcholine for future use. So that's how we can terminate the contraction event, close those channels. Now you can see what they look like when they're not bound by their ligand. Okay, so we'll back out a little bit and see what the big picture looks like, at least bigger picture. Sodium ions are entering through these open acetylcholine receptor channels and depolarizing the neuromuscular junction. Those ions then diffuse to the perimeter of the neuromuscular junction. And in the perimeter of the neuromuscular junction are two types of membrane protein channels, a sodium channel and a potassium channel, both of which are electrogenic, electrically sensitive or gated. These channels remain closed whenever the interior is negative, but when the interior becomes positive or depolarized, they open. All right, so we have sodium ions diffusing to the perimeter, and as a result, the first sodium electrogenic channel will open, and sodium ions will then flood into the cell. And as sodium ions flood into the cell, they will depolarize the region, and the next sodium electrogenic channel will open. And these channels reside on the entire surface of the muscle cell. So this wave of depolarization will spread across the entire cell membrane with no way to stop it. That's called an action potential. Sorry, I'm in the way of everything. An action potential is this wave of depolarization that spreads automatically from channel to channel across the membrane. It's like what's happening in California right now, right? If someone drops a match anywhere, it'll catch fire on something and it'll start a fire that will burn thousands of acres. Well, we're gonna depolarize acres of muscle cell membrane here. So here's another way of representing those channels. As the membrane depolarizes in the perimeter of the neuromuscular junction, an electrogenic sodium channel opens. These little trap doors are repelled by the positive charge now as the membrane depolarizes, and they flip up, open the channel, and sodium ions rush in and further depolarize the membrane. And then those ions diffuse along the surface of the membrane and depolarizing it. So the pink arrow represents the progress of sodium ions diffusing along and depolarizing the membrane. And we have to imagine more sodium channels positioned all along the membrane. Excuse me. Over here we can see part of the membrane that still has the resting membrane potential. So this is a dynamic thing. It's moving down the membrane like a wave. That's called propagation or spreading. That term is kind of going out of vogue. So the action potential spreads along the membrane from sodium channel to sodium channel. Well, only a short while later, the sodium channels inactivate. They essentially jam up. This little ball is meant to represent a change in the state of the sodium channel. It can no longer let any more sodiums in. It has not gone back to its resting state. You can see that the trap doors are still open, but there's something in the way and no more sodium ions can rush through. So it's stopped. At the same time, some electrogenic potassium channels start opening. They're responding to the same initial depolarization as the sodium channels. They just physically take longer to open. And so during that delay, we have a wave of depolarization, and then all of a sudden the potassium channels start to open. So when the potassium channels open, they let potassium ions, positive charges, out of the cell, which then leaves behind, again, that net negative charge inside the cell, repolarizing the membrane. Going back to the resting potential, right? Negative charge interior, positive outside. And that wave spreads. These negative signs here represent the progress of the potassium ions being let out of the cell, and then we go from potassium channel to potassium channel. Again, we have to imagine potassium channels along the whole surface of the cell. Another way of visualizing the action potential is to think what it would look like if we could stick a tiny wire into the membrane at some point, and a tiny wire outside the membrane in the fluids, and connect them to a voltmeter that measures charge differences. That's what a voltmeter does. And as the action potential spreads across the membrane, the wave of depolarization, and crosses the point where our voltmeter wires are, we would see an increase in the charge on the membrane, going from a resting potential, a negative resting potential at our voltmeter, and as the wave of depolarization sweeps by, we see the membrane getting more and more positive as sodium ions rush into the cell. That's called depolarization, as you know. It could reach as much as plus 30 millivolts momentarily at that point, and then the sodium channels will inactivate, no longer let sodium ions in, and potassium channels, the so-called slow potassium channels, responding to the same initial depolarization down here from the neuromuscular junction or from a neighboring channel, a neighboring sodium channel, begin to open and let potassium ions out. And as positive charges are leaving the cell membrane, the membrane starts getting more and more negatively charged, repolarizing the membrane until we get back to rest. And eventually the potassium ions close, the sodium ions reset themselves, and we can do it again. And every time there's an action potential, there's either, if it's a neuron, there's more calcium ions rush into the terminal. If it's a muscle cell, this wave of depolarization will spread along the membrane. So that's just looking at one spot as this dynamic sequence of events passes by our viewing point. So now let's back out and look at the big picture a little bit more so we can see what's going on in the muscle cell, because this is the way we activate a muscle contraction. So the action potential came down the axon to the terminal, depolarized the terminal, calcium ions went in and exocytosis of acetylcholine took place, depolarized the neuromuscular junction by the opening of receptor channels. That spread to the perimeter, and then electrogenic sodium channels began to open, and a wave of depolarization spreads across the entire cell membrane and down into the T-tubules. The transverse tubules are part of the cell membrane. So the wave of depolarization spreads down deep into the muscle cell, snuggled up against the terminal cisterns of the sarcoplasmic reticulum. So when the wave of depolarization enters into the T-tubules, there are electrically sensitive proteins in the membrane there, called foot proteins, that change shape or conformation when the membrane depolarizes, and they in turn change the conformation or shape of some valves, some channels on the membrane of the terminal cisterns, and the sarcoplasmic reticulum lets calcium ions shoot out into the cytoplasm like a lightning flash. So depolarization waves hit, calcium ions are released. So now we're getting somewhere. Calcium ions are the switch that activates the contraction of the muscle. So as the calcium ions enter into the cytoplasm around all those myofibrils and all those sarcomeres of the myofibrils, calcium ions bind to troponin, one of the regulatory proteins we saw. Right at rest, the two regulatory proteins keep the actin filaments blocked so that myosin heads can't bind. There's a sheath of tropomyosin blocking all those binding sites. Calcium ions then bind to troponin, and troponin changes shape like a handle moving the tropomyosin, and now we can see all those dark spots which represent myosin binding sites are exposed, and immediately the myosin heads will bind to the actin filaments. And once they bind, it's like pulling the trigger, and there's a spring-loaded hinge there that just rotates, and that myosin head actually pulls the actin to the left and the myosin to the right in this illustration, and the two filaments physically move. That's how muscles contract. Taking chemical energy, right, the original source of energy was the sodium-potassium pumps, and some ATP, as we'll see in a second, in the muscle cell, but sodium or calcium ions rushing in activate this conversion of electrical energy into mechanical energy or work. Sorry, I'm blocking everything. All right, now, is that the end? That would only make a tiny little molecular-sized contraction of a muscle, but much more than that happens, because once that myosin head rotates, it becomes ready to repeat itself. It's a cyclical process of myosin heads grabbing actin and moving it. Let's take a look. So here's our myosin head that has just rotated, like the spring being released and moving the actin to the left and the myosin to the right. Once the head changes position, the myosin loses its affinity for two molecules that were bound to it, adenosine diphosphate, or ADP, and inorganic phosphate. It's a phosphate with 4 oxygens. Well, the minute those things are released, that changes the properties of the myosin head. Oh, I'm sorry, no, that's not the case. ATP can then bind. It changes not the affinity, but well, I should have kept going. It changes the affinity for ATP, and ATP can bind to the myosin head, which then lowers the myosin affinity for actin, and it dissociates. So let me clean up that little bungle. ATP binding to the myosin head is required to dissociate the head from the actin. Once that happens, the myosin exhibits ATPase activity. It is indeed an enzyme, and it hydrolyzes the ATP, meaning breaks off the inorganic phosphate. That's the high-energy phosphate, and releases a bunch of energy, and that released energy resets the spring-loaded head, myosin head, into the cocked position, and it's like having a latch. There's some tension there when it's in that position. Then, once it's in that new position, the affinity or stickiness for actin is restored, and the myosin head grips onto the myosin binding site on actin filaments. And the minute that happens, the binding changes the myosin, and pulls the trigger, releases that spring tension, and it rotates and does some work and moves the actin. So around and around and around we go. As long as there's calcium in the cytoplasm, see the calcium ions bound to the troponin, and plenty of ATP around, the cyclical cross-bridge rotation, it's called, will continue. And we'll just keep moving the actins so that the sarcomeres will shorten and shorten, and the muscle will contract. How will this ever end? It will end when we stop releasing acetylcholine. Acetylcholine esterase will break down acetylcholine in the neuromuscular junction in the synapse. The membrane potential will go back to rest, and the calcium ions will be pumped back into the sarcoplasmic reticulum by calcium pumps, another type of primary active transporter. In the sarcoplasmic reticulum are ATP-using calcium pumps that pump the calcium very quickly right back into storage in the sarcoplasmic reticulum. And when that happens, then this process will stop. We'll be back into this position where the actins are blocked by a sheath of tropomyosin. There's no more calcium out here to bind to the troponin, and the muscle cell will be at rest. So there's just a couple slides here summarizing all these steps as I've just done looking at the picture. Action potential, the impulse, travels down the motor neuron once we decide to do a contraction. It reaches the terminal at the neuromuscular junction, and acetylcholine is released by exocytosis. The ion permeability of the muscle cell membrane, the sarcolemma, changes, namely because the acetylcholine receptor channels open. Sodium rushes in and depolarizes the muscle cell membrane at the neuromuscular junction. That in turn depolarizes the perimeter, and that ignites the action potential, right? Once the depolarization reaches the perimeter of the neuromuscular junction, the electrogenic sodium channels open and a wave of depolarization starts. And it spreads across the entire cell membrane down into the T-tubules, from sodium channel to sodium channel to sodium channel, and then eventually a wave of repolarization. And when the depolarization affects the T-tubules, the sarcoplasmic reticulum will be induced to release its calcium by opening up its channels, and will have calcium triggering the cross-bridge cycle and the contraction of the muscle cell. All that process is called excitation, contraction, coupling. Once we trigger the action potential in the cell membrane, all those events leading up to the cross-bridge cycle are called excitation, contraction, coupling. Right? This is the excitation phase. This is how we convert that into actual mechanical events in the cell and shorten up that muscle cell and do physical work in the environment. So that's pretty miraculous. That's the claim to fame of muscle cells. They can convert chemical energy in the form of ATP into mechanical work. So remember, we're using up an ATP every time every cross-bridge cycles, and thousands of cross-bridges are activated, or myosin heads are activated in cross-bridge cycling and so forth. So that's how we convert chemical energy into mechanical work. Okay, next time we'll carry our story further and start looking at the bigger picture of muscle physiology. Where does the energy come from for all that ATP for muscle contraction? And how do we regulate, how do you determine how much tension is going to be produced by muscles? You know that you can gently contract a muscle and lift up a very lightweight object, or you can lift up something very, very heavy using the same muscles. So a number of interesting things we'll be looking at in part 3. So join me for that.